Hi, everyone. My name is Jing Liu. I am a member of the bioinformatics team here at Myriad. I'm a bioinformatics analyst here. And today I will be talking to you guys about my work on my project called DeepCAH or DeepCAH. You can call it either one. And its involvement in deep learning and variant calling for difficult genes. So, since this is one of my first Bay Bifics events, I figured I should more properly introduce myself. I graduated from Duke University last May, studying biology and computer science. Shortly after I graduated, I became the bioinformatics intern here at Myriad, where I started my work on DeepCAH. And this difficult gene variant calling. So I primarily worked on DeepCAH during my time here as an intern, and recently I converted full-time and I'm now a bioinformatics analyst here. So super excited to be here as a full-time employee. And yeah, I'm just continuing my work on DeepCAH now along with some other things. So yeah, that's a brief intro about me. So before I get into what deep CAH is, I wanted to talk about what CAH is. So CAH stands for congenital adrenal hyperplasia, and it is a group of genetic disorders or conditions that affect how the adrenal glands, which are these glands over here sitting on top of the kidney, produce steroid hormones. So the adrenal glands are responsible for producing hormones such as adrenaline, epinephrine, norepinephrine, cortisol, like all these kinds of things. So they're very important to the human body. And it is because it's a group of genetic conditions, it is very highly variable between individuals that do have CAH. And CAH affects 1 in 10 to 20,000 individuals in the general population. So it's not super common to have CAH, but it's not exactly entirely super rare either. However, it's important to note that in the general population, 1 out of 50 to 75 people actually have at least one copy of a CAH version of the gene, which is why Myriad has historically offered CAH on their foresight assays, which is their carrier screening assays. And yeah, as I mentioned before, CAH is very highly variable. However, CAH, the vast majority of CAH cases involve a loss of function in the gene called CYP21A2. So this gene is responsible for producing an enzyme called 21 hydroxylase. So when you get a loss of function mutation in this gene, you end up getting a 21 hydroxylase deficiency. Um, so if we see this slide, again, CAH is super highly variable between individuals. So this is kind of a general sense of how CAH can affect an individual that has CAH. And basically, you can see like there's a loss of function in the gene, in the target gene that we're talking about here. And with this loss of function, there's less cholesterol that is being converted into cortisol and aldosterone. And therefore, there's more cortisol left over to be converted into estrogen and testosterone. So you can see how it could affect someone with CAH. There tends to be like a hormonal imbalance as a result. So now let's jump into the genetics behind CAH. So CAH is very notorious in the bioinformatics community to be very difficult to genetically analyze. And one of the primary reason behind that is the genetic complexity of CAH itself. So I mentioned the gene CYP21A2. This gene is actually sitting next to a pseudogene, which is not functional, so it doesn't actually produce the enzyme that I mentioned earlier, but is 98% identical to the gene in terms of its sequence. So that can be kind of difficult to navigate. And on top of that, there's also tons of polymorphisms and recombinations between the gene and the pseudogene. So with these like little illustrations that I have at the bottom of this slide, you can see how there are many ways that the gene and the pseudogene can be recombined, which makes it just overall a very complex problem. Genetically speaking. And because of this complexity, it raises a lot of frustration from our lab directors here at Myriad. So what the lab directors do is that they take the results of the CAH legacy caller, which is basically this more rudimentary variant caller for CAH, excuse me, that doesn't use deep learning, and they have to manually review all the CAH calls, which, as you can imagine, is super, super time consuming and takes up a lot of their time and energy as lab directors. And on top of that, when a call is reported incorrectly, that obviously is very problematic to whatever patient is involved. So whenever that does happen, they have to go back and report the error and explain why the error happened and how to prevent it. So that entire process, or a kappa, is also very time consuming in addition to the manual reviewing of these CAH calls. So this is where DeepCAH comes into play. So DeepCAH is a deep learning based model that helps the lab directors conduct variant calling for CAH. So basically how it works is that it takes in some input from sequencing data. So in our case, it would be SNP ratios, copy number information, SNP information, et cetera. And it gets inputted into this ML model, and the ML model essentially spits out these variant calls. So we have a preset list of variants that we're looking for, so like variant 1, 2, 3, etc. And the ML model spits out results for whether or not a given sample is positive or negative for each of these variants. One important thing to note is that this machine learning model can only detect variants that were already preset, so we can't call basically new variants, if that makes sense. So here's a general overview of what the DeepCAH pipeline looks like. It kind of looks like a lot, but we can split it into two halves. So the top half, the blue half, is the training half, and that's the basically training pipeline that ends up producing the machine learning model. So what we do is that we take in an input CSV data set that includes the SNP and copy number information that I mentioned earlier, as well as reported calls that were already determined by lab directors. And these reported calls will serve as a sort of ground truth for the training to work upon. Once we have this input CSV data set, we input it into the training app, and it kind of like cooks for a little bit, and it outputs a machine learning model. Once we have this machine learning model, we can then use it for inference. So yeah, there are a lot of boxes here for inference, but the main takeaway is that when we have an input CSV data set with just SNP and copy number information, we can put that into the machine learning model that we just trained and then create inferences based for the samples based on the copy number and SNP information. And eventually these inferences then get sent to the lab directors who will then review it just once again to make sure everything's correct. So there's actually an older model of DeepCAH that was previously at Myriad prior to me joining. So when I joined Myriad as an intern, there were some things that we wanted to modify, update, about this old model. So there are many similarities. For example, it still retains the deep learning aspect of it all, and the input data overall looks the same. However, there are some main differences. So the first one is updating the ML architecture. ML has progressed quite a bit since it was last touched, since DeepCAH was last touched. So we wanted to update the ML architecture to make it more modern. We also wanted DeepCAH to be compatible with newer training data. We also modified the preset list of variants that we wanted to modify the preset list of variants that we are trying to detect using DeepCAH. In this case, we only added about like 1 to 2 variants, so nothing too crazy, but we just modified it a little bit. And finally, the lab directors did request us to implement zygosity detection. So what that means is that instead of having just like a binary positive negative call for each variant, we're going to have calls that look like 0, 1, 2. And the number essentially represents the number of copies of CAH present, if that makes sense. So the overall goal of the new model is that we wanted to maintain the same level of high performance as the old model, except make it compatible with newer data and incorporate zygosity calls as well. So if we go into the architecture, so there are many ML architectures that we decided to investigate. The first one being long short-term memory, or LSTM. So LSTM is the architecture that the old model originally used, and it has been shown to be good with sequential or time series data, which makes sense because a lot of genomic data is sequential in nature. So that was kind of their reasoning for using LSTM. However, looking at the data set myself, a lot of the data set was basically like a large like CSV spreadsheet with a bunch of decimals in it. So it didn't really match the sequential nature of the data that LSTM was suited for. So we decided to explore other options, which you can see on the right. There are 3 more architectures that we considered. And the main takeaway from these architectures is that all 3 of them seem to be really good with the type of data that we were using for DeepCAH. However, after many rounds of training and retraining and analyzing ML metrics, we ultimately decided that LessONet was the best option to move forward with our new model. And so here's a plot of the different metrics between the old model and the new model. And we wanted to see whether or not the new model held up with the performance of the old model. And as you can see, like, it does like pretty similarly. There's some metrics where the new model does significantly better than the old model. Sometimes the other way around as well. But overall, it does a pretty like similar high performance job compared to the old model. And so after we have this trained DeepCAH model, we can then use it for inference. So once again, I'm just going to reiterate. So we can prepare a new input data set and then put this input data set into our newly trained model to then produce inferences and send it to the lab directors or LDs for review. So, what are our next steps for DeepCAH? So before we get like too excited and incorporate our new model into the pipeline, we wanted to first analyze how DeepCAH handled complex cases. And what I mean by complex cases are, for example, samples with multiple positive calls, because DeepCAH historically has been a little bit iffy, I would say, about samples with more than one positive call. So we wanted to make sure that DeepCAH performed just as well with those cases as well. And there's also new data coming into the ARMS database. So the ARMS database is the clinical data report database that we use. And because there's new data coming every single day, we want to eventually collect all this new data to then train another model, because as we know, like the more data for machine learning, the better. And once we have this model, we want to run inference based on the slide that I mentioned earlier and incorporate it into our pipeline. And once this is finally incorporated into our pipeline, we want to re-include it into our foresight assay. So then when we sell the foresight assay to customers, they can now test with CAH. So that will be very exciting, but that'll be down the road. So I like to shout out everyone on this slide. Special shout out to Sam Perlman, who was my intern mentor for the course of my internship. And he has continued to help me with DeepCAH after I converted as a full-time employee. Shout out to Shane Brubaker as well for organizing this event and also letting me speak here to talk about my work. And yeah, thank you guys for coming, and thanks.